Hi everyone! On this video, we from IUP class group 4 are going to explain about protein synthesis. Our group member consists of Menzavadian Amril, Salwa Alia Putri, me, Anindiana Rimaza Rashidina, and also Akilah Dia Ulhaq. Protein synthesis is the process in which cells make proteins. It occurs in two stages, transcription and translation. The process by which information in genes flows into proteins is DNA to RNA to protein. It is called central dogma of molecular biology. Now we are going to talk about RNA. RNA is made of nucleotide consisting of a 5-carbon sugar ribose, a phosphate group, and a nitrogenous base. It carries out the instructions coded in DNA. There are three types of RNA. The first one is messenger RNA or mRNA. It carries information from DNA in the nucleus to ribosomes in the cytoplasm. The next one is ribosomal RNA or rRNA. It is a structural component of ribosome. And the last one is transfer RNA or tRNA. It carries amino acids to the ribosomes during translation to help build an amino acid chain. The differences between RNA and DNA are: the first one, RNA uses sugar ribose instead of deoxyribose. The second one, RNA is generally single-stranded instead of double-stranded. And the last one, RNA contains uracil in place of thymine. The four bases of RNA form a language with just four nucleotide bases: adenine, cytosine, guanine, and uracil. The genetic code is read in three base words called codons. Each codon corresponds to a single amino acid or signals the starting and stopping points of a sequence. There are 64 possible codons. As you can see in the slide, in addition to instructing the addition of a specific amino acid to a polypeptide chain, three of the 64 codons terminate protein synthesis and release the polypeptide from the translation machinery. These triplets are called Nosen codons or stop codons. Another codon, AUG, also has a special function. In addition to specifying the amino acid methionine, it also serves as the start codon to initiate translation. The reading frame for translation is set by the AUG start codon near the 5' end of the mRNA.
Okay, next is transcription. The transcription is the first step in gene expression, in which information from a gene is used to construct a functional product such as a protein. The goal of transcription is to make an RNA copy of a gene's DNA sequence. For a protein coding gene, the RNA copy or transcript carries the information needed to build a polypeptide, protein, or protein subunit. Eukaryotic transcripts need to go through some processing steps before translation into proteins. The main enzyme involved in transcription is RNA polymerase, which uses a single-stranded DNA template to synthesize a complementary strand of RNA. Specifically, RNA polymerase builds an RNA strands in the 5 to 3 direction, adding each new nucleotide to the end of the strands. Transcription of a gene takes place in three stages: initiation, elongation, and termination. Initiation: RNA polymerase binds to a sequence of DNA called the promoter, found near the beginning of a gene. Each gene, or group of co-transcribed genes in bacteria, has its own promoter. Once bound, RNA polymerase separates the DNA strands, providing the single-stranded template needed for transcription. Second one is elongation. One strand of DNA, the template strand, acts as a template for RNA polymerase. As it reads, this template one base at a time. The polymerase builds an RNA molecule out of complementary nucleotides, making a chain that grows from 5 to 3. The RNA transcript carries the same information as the non-template or coding strand of the DNA, but it contains the base uracil U instead of thymine T. Last one is termination. Sequences called terminators signal that the RNA transcript is complete. Once they are transcribed, they cause the transcript to be released from the RNA polymerase. An example of a termination mechanism involving formation of a hairpin in the RNA is shown below. In bacteria, RNA transcripts can act as a messenger RNAs or mRNAs right away. In eukaryotes, the transcript of a protein coding gene is called pre-mRNA and must go through extra processing before it can direct translation. Eukaryotic pre-mRNAs must have their ends modified by addition of a 5' at the beginning and a 3' at the end. Many eukaryotic pre-mRNAs undergo splicing. In this process, parts of the pre-mRNA, called introns, are chopped out, and the remaining pieces, called exons, are stuck back together. End modification increases the stability of the mRNA, while splicing gives the mRNA its correct sequence. Not all genes are transcribed all the time. Instead, transcription is controlled individually for each gene, or in bacteria for small groups of genes that are transcribed together. Cells carefully regulate transcription, transcribing just the genes whose products are needed at a particular moment. For example, the diagram below shows a snapshot of an imaginary cell's RNAs at a given moment of time. In this cell, genes 1, 2, and 3 are transcribed, while gene 4 is not. Also, genes 1, 2, and 3 are transcribed at different levels, meaning that different numbers of RNA molecules are made for each.
The next one is translation. Translation of the nucleotide sequence in the mRNA molecule into the amino acids that make up a polypeptide. It occurs in ribosome. For the mRNA, it is a template. For the tRNA, it brings the amino acid into ribosomes. The steps of translation, the first one is the activation of amino acid. The carboxyl group activated and then facilitate formation of peptide bond, and then each amino acid attached to a specific tRNA using amino acid, and then the tRNA synthesis, and then the tRNA charge. It takes place in cytosol. The next one is the initiation stage. In this stage, the ribosome gets together with the mRNA and the first tRNA, so translation can begin. And then the tRNA binds with methionine to start codon to the small ribosomes unit, forming initiation complex. And then the initiation complex will move until AUG. The third stage is elongation. This is the stage where the amino acid chain gets longer. The anticodon in tRNA forms a base pair with the codon in mRNA. And then the catalytic site on the large subunit catalyzes the formation of peptide bonds connecting the first and second amino acids. And the last stage is termination. Termination happens when a stop codon in the mRNA UAA, UAG, or UGA enters the A site. The protein synthesis terminates once a stop codon has been encountered. The new polypeptide chain is released with the help of termination sequences.
Now we move to the mutation. When an organism or cells contain 2n chromosome or more other multiple of n complete, it is said to be haploid. If a cell is missing one or more chromosome, it is said aneuploid. Two common types of aneuploid have their own special name. The first one is monosomy, is when an organism has only one copy of chromosome that should be present in two copies, like 2n minus 1. The second one is trisomy, is when an organism has a third copy of a chromosome that should be present in two copies, like 2n plus 1. An haploid has also include cases where a cell has a large number of extra or missing chromosomes, as in 2n-2, 2n plus 3, and others. However, if there is an entire extra or missing chromosome set, this is not formally considered to be unemployed, even though it may still be bad for the cell or organism with more than two complete cells of chromosome are said to be polyploid. The most common trisomy among embryos that survive to birth is Down syndrome and trisomy 21. Disorders of chromosome number can caused by non-disjunction, which occur when a pair of homologous chromosome or sister chromatid fails to separate during meiosis 1 or 2 or during mitosis. Non-disjunction can also happen during mitosis. In humans, chromosomes change due to non-disjunction during mitosis in body cells will not be passed on the children because their cells don't make a sperm and egg. But mitotic non-disjunction can cause other problems, like cancer cells, often with abnormal chromosome numbers. When an unemployed sperm or egg combines with a normal sperm or egg in fertilization, it makes a zygote that is also unemployed. For instance, if a sperm cells with one extra chromosome combine with a normal egg cell in result zygote, or one cell's embryo will have a chromosome numbers of 2n plus 1. In another class of large-scale mutation, big chunks of chromosome but not entire chromosome are affected. Such changes are called chromosomal rearrangement. There include the first one, a duplication, when a part of chromosome is copied, the deletion, when a part of chromosome is removed, an inversion, when a chromosome region is flipped around so that it points in the opposite direction. The last one is translocation, when a piece of one chromosome gets attached to other chromosome. A reciprocal transition involves two chromosome swapping segments. A non-reciprocal traction means that a change of one chromosome moves to others. That's all from our group. Thank you so much for watching.